and we are going to have an expert panel here. If you want to know, I'm not even sure if I prepared slides. I think, do you have your PowerPoints ready to go? Okay, so this is a challenge. The size of the gene is a major issue for RYR-1. It's also a major issue for Duchenne's. And so what we have done at the foundation is looked outside of the field of RYR-1 to see, well, what's going on in other fields like Duchenne's that have this issue. And the most recent round of research grants are all funding that. And if you want to know how your dollars are being put to work, if you're a donor to the foundation, you can look right here. We are funding their work, and all of their work is looking to overcome and mitigate that issue related to the gene size. Each one of these people have different strategies to overcome it. And what you're going to hear is some of the most cutting-edge, latest research that's going on that is very, very promising. They all have benefits, they all have challenges, and we're going to work through that. But it's really exciting, and it's really exciting to see foundation dollars at work to support this really important research. Afrouz, I think you're up first.
So I'm sure you heard a lot today about the genes and how the mutations can cause this RYR-1 disorder. But I wanted to start with few slides that helps everybody to explain their gene therapies. The first one is the central dogma of biology, and it's basically tell us that all the information in our body, that we call it DNA, comes from our parents. And this is an instruction for making a messenger we call RNA, and then this RNA will be translated into the protein, that there are small, tiny machines in our body that they will make the entire body of each person, from our hair, skins, muscles, nerves, everything. And if there is any issue with this information we're receiving from our parents, in our DNA, finally our proteins will be defective and they will not perform the role that they're supposed to do in our body. So what gene therapy does actually works on a DNA. So we transfer a healthy copy of that defective gene to patient cells to make a healthy protein to perform all the functional things in the body. And we are using the adeno-associated viral vectors for transferring this healthy copy of the genes. So here I just want to show you how tiny these AAVs are compared to other small things. For example, they are about 8,000 times smaller than hair, or about 4,000 to 6,000 times smaller than a sand beach, like a tiny thing, and like 4,000 times smaller than a tiny salt piece. Or even compared to the COVID virus that caused an issue in healthy people, they are about 50 times smaller than a COVID virus. So they are so tiny, but because they are non-pathogenic, they don't cause any diseases in human. So they have been chosen to deliver a healthy copy of our genes to the patients. So, and there are here just, I try to draw a circle around them so you can find them easily, but they are so tiny, tiny dots. They are like a dust on the dust, so they are very small. But as they are not pathogenic, they have been chosen vehicle for transferring some healthy genes. And already FDA approved about 7 of these AAV-based gene therapies for treating different kind of disorders, including eye disorders, muscle disorders like Duchenne, or for neurological disorders like SMA, or liver disorders and metabolic disorders. So I just listed them here. And you can imagine these AAVs as a Trojan horse. So I'm sure everybody heard about the Trojan story, that it was a about 3,000 years ago, a war between Greek and Troy city, and it took for 10 years. But finally, Greeks came with a very bright idea of making a gift for Troy people. It was a wooden large horse, but they put their soldiers inside that horse and they give it to the city as a gift. And at night, those soldiers came out and opened the gates. So the Greek soldiers came in, and the war has been ended by Greek vote. So these viruses are really like this Trojan horse. They can be seen as harmless thing through our cells, but when we put our healthy genes inside them, when they are in our body, they can express the healthy proteins. So we use these adeno-associated viruses for other disorders in our lab, but for RYR1, it has been challenging because, as it has been told today several times, the RYR1 gene is so big. So we need to cut it into 4 pieces and place it into 4 AAV vector, and then inject all of them together to the patients to produce the full length RYR1 gene and protein. The strategy that I'm working on, and the grant has been funded, it's called RNA end joining. So you can imagine how sewing a dress can happen by attaching small pieces of the fabrics together to create a dress. So actually here you can imagine those 4 pieces of RYR1 RNA that we talked about as these pieces. And when they are inside the body, our cell will be acting as a very a very good, let's say, tailor, and just sewing these pieces together and make the full length RYR1 that is functional in our body. So this is the strategy that I'm using in the lab, and we received the funding from RYR-1 to continue the research. So by that, I just want to thank everybody in the lab who works on this research, and actually RYR-1 for funding this research, and all of you for supporting our research.
Any quick questions before we move on to the next speaker? All right, Afrouz, thank you so much. We'll have some more questions and answers sessions. Celine, is Celine next? Celine. All right, Celine, our sole speaker from Pittsburgh.
Acquired.
Have to pay for airfare. We're very happy about that. All right, welcome, Celine.
Yes, don't look at my accent. So I'm native English speaker from Pittsburgh. I'm from Italy, but I was trying to make a joke. I think it did not work out properly. But yes, so I'm very happy today to give you a little talk about herpes simplex viruses and how to use them as gene therapy tool for RYR1. Now, I'm trying to simplify this as much as I can. Now, herpes, I think everybody knows about him. Imagine to strip the virus from all the viral genes that cause cytotoxicity. And so you go from a wild-type virus to a virus that becomes just a vehicle. Similar to AAV that you have heard many times, or lentiviruses. HSV is currently moving forward to clinical trials. So I wanted to show you just a couple of trials. So once we stripped the virus from these viral genes and added our gene of interest, the current application that we can find are an FDA-approved drug for wound healing, a clinical trial for cystic fibrosis, another clinical trial for bladder disease, and one for pain. So little by little, HSV is taking his place. I think I'm going to be fast here because you have learned this over the course of this morning. So when RYR-1 is a channel, and when it's functional, goes from a closed to open. And so what happens, though, when this mechanism fails? So there is, as you have heard from all the experts this morning, there are several heterogeneous mutations that can occur in this channel. The one I will work on in this grant is about recessive mutations, so the one that reduce the amount of protein in the body. So what I wanted to say, this entire mechanism, as it was explained before, is extremely complex. So, and this is due to the huge, the gigantic, large size of this gene. So to deliver it all, you need a big delivery system. So imagine it as a puzzle. You want to deliver all the pieces, right? You're not going to gift a kid with one piece only. So there is like different vectors and different capacity for these vectors. So AAV, as you have heard, is used in clinic, but it's a very small vector. So he can deliver pieces, and he can deliver multiple pieces if you have multiple vectors. Lenti can deliver a little bit more. HSV can deliver it all. So the idea of this project is to create this HSV-based vectors, have the entire RYR-1 gene in this vector, and study that, as Dr. Marty said before, first in muscle cells, and then deliver it in a mouse model. And we hope to deliver, our goal is to deliver the functional RYR-1 gene safely back into muscle with gene therapy. And with this, I will conclude, and I want to thank everybody and the RYR-1 Foundation. Thank you so much.
Any questions for Celine? So, all right, Celine, just to be clear, the advantage of herpes simplex virus over AAV is the size of that it can contain the entire RYR-1 gene. Yeah. And just to be clear, because I suspect there are some people out here worried about this, if you were to get this viral vector, there is zero risk of genital herpes. Yes. And oral herpes.
Yes, there is no risk.
Just want to, I know there's some nervous laughter out there. I know who you are. No, there's no risk.
No, there is no risk.
Okay.
The virus is incapable of replicating. It's just a carrier right now.
Okay, and last question is, how good is it at targeting muscle relative to liver? So we've talked a little bit about some of the liver issues that have been seen in the AAV trials. Do we know any liver issues associated with herpes simplex virus used in this application?
No. So the clinical trials currently have not evaluated liver toxicity. Most of them were for cystic fibrosis directly inhaled.
Okay.
For wound healing is applied by a cream on the wound. Pain, so it's not delivered IV intravenously yet. Okay. In the future, we hope to modify the envelope to redirect the virus specifically to muscle cells, and that's possible. It's the next step, hopefully, so that we can deliver it IV.
Yeah, and so what's going on in gene therapy is trying to, as I mentioned earlier, trying to get the vector or the delivery vehicle to really have a high level of attraction to muscle that allows you to lower the dose and allows you to avoid some of these liver issues. That has occurred to some extent with AAV, and it sounds like you're working on that with HSV, which is really exciting. So, Celine, thank you so much. Round of applause for Celine. All right. So we saved this is all complicated stuff, and I'm still trying to wrap my head around it, but if Haichum can explain this topic in a way that we can understand, this will be the Nobel Prizes. I mean, I'm calling the Karolinska Institute. But this is a really exciting, these are all exciting projects. This is also great. HiGym has done great work in this area, again, initially on Duchenne's, and now we're looking at RYR-1. HiGym, could you maybe briefly talk about your background prior to this with CNM and Dr. Laporte, and your sort of, you had some level of understanding of RYR-1?
Sure. So, hi everyone. First of all, I'm really happy to be here. You can't imagine how happy I am today to join you. Yesterday we had a great scientific meeting. I think I'm more stressed and more excited to come and talk to patients and their families. I'm an assistant professor now in the Department of Neurology in the University of Washington in Seattle. During my postdoc, I worked on Duchenne muscular dystrophy, which shares some common features and challenges to RYR1 related myopathies. But I was trained and I got my PhD in France, where I worked on centronuclear myopathies. And as you heard this morning, centronuclear myopathies is a big family, and RYR1 is one of member of these. So I've been working on RYR1, but on other diseases like myotubular myopathy that Isabelle Marti presented a few minutes ago. And now I am trying to apply the technologies that I learned during my postdoc that we applied to Duchenne muscular dystrophy to RYR1 because they have some common challenges. Is that good? Okay. So let me reiterate some few points. I know that presenting the last slides is a little bit, everything was said before brilliantly by the other experts. So the genetic, in many genetic diseases, they have the same issue, which is small typos in the DNA, which is the instruction manual to the cell to work correctly. And these genetic diseases, they block or they make some errors, and then the protein that is really important for functional cellular function are impaired and are not correctly produced. So there are a couple ways to mitigate this, either to correct the mutation using CRISPR-Cas9 that Isabelle Marti just presented a few minutes ago, or use AAVs or other different viral vectors to deliver a healthy copy that we know that will function correctly in cells. And this will help to restore the production of this missing protein, and this is what we call gene replacement. So with gene replacement, we replace the faulty gene by a correct copy using a gene carrier or transporter into the affected cells. So different vectors are being developed and used. You heard AAV with the Fruz, and also HSV previously. So in my lab, we are using the same AAVs, so adeno-associated viral vectors, which have been used and shown promising results in preclinical and clinical studies. More than 200 clinical trials are now ongoing, and they are very efficient in delivering this correct copy to the affected cells. And as you heard this morning, and a few minutes ago, this AAV vector is small. It can't fit all the genes that we want to deliver. So that is the challenge. And this poses an enormous challenge to deliver large proteins like RYR1. So I come from outside of the US. We have small cars. I have been in this situation myself, where I go to buy a piece of furniture and I get stuck. So I was just praying if I can find a way to, deassemble this piece of furniture and then put it inside my car. So that's the idea that we want to do with RYR1, is to fragment this protein into pieces that can fit in the viral vector and then send it to the affected muscles. So the way we are doing it is that we are using these new technology called splitting. So the idea is to fragment this RYR1, put these pieces that can fit into the AAVs, and then combine these 4 vectors that are carrying these pieces of the RYR1, inject them into the body, and then these AAVs, they travel in the bloodstream, they go into the affected muscles, they start producing these pieces, and then the trick here is that we are using a few small elements that you can see here colored in yellow, green, and purple, which act as a glue. So these will fuse these different fragments into one fragment. So from 4, we will go into produce one large protein, which is the RYR-1. And this is extremely efficient. We did it and we validated it in other diseases like Duchenne muscular dystrophy and other muscular diseases. And now we are transitioning to RYR-1. So it's going to be challenging, of course, but I think we have very promising data that we obtained from cells using plasmids, and now we are producing these AAVs, and thanks to the RYR-1 Foundation, so we got money to fund and get this project going. So the idea is to produce these AAVs, 4 of them. So we just finished making them making the DNA that is needed for the production of AAVs. And the idea now is to test these AAVs into cells that are derived from patients, RYR-1 patient, and test the efficacy and the feasibility. Is it doable to do this approach? And then once we have this data, then we can move to test it in mouse model, as Isabelle Richard mentioned, because Isabelle Marty, mentioned. So because we need to have this approach validated in a mouse model and before going to patients. And this is what will be funded by the RYR-1 Foundation. And with that, I would like to thank the people in my lab, also partners, RYR-1, MDA, AFM Téléthon, and the Foundation Betancourt. And also you see here that I'm mentioning the TC family, who gave us $20,000 before submitting this grant into to the RYR-1 Foundation. So with $25,000 we could generate preliminary data and could convince the scientific advisory board of RYR-1 that this technology can work for RYR-1 like with Duchenne muscular dystrophy. With that, thank you very much for listening.
All right, thank you. Hi, Jim, thank you so much. Any questions? Oh, wait. My mother. Here we go. Get comfortable. She has a lot to say.
How do I get this to work?
It's working.
Wonderful. So I love your illustration of the sofa in back of the car. And, we can picture that. But it has to be in pieces to get in the car, right? And then it's got to be put back together. So when these 4 sections of genes have to get back together, what's going to prevent one of them from getting lost on the way?
That's
or
a good
finding
question.
the other,
Yeah.
or another one gets lost. 2 of them get lost.
Yeah.
And wait, they don't know where they are. How are they going to find each other?
So that's a problem that we call biodistribution. We are using a highly potent vector. So we are using vectors, they know how to go to muscle because they were engineered in labs to go and target specifically and efficiently skeletal muscles. And then in the cell, how they know and where to find each other, I don't know, but it's working. So we're still trying to understand how this mechanism is working, but it's a great question. So we don't have data on how these find each other. But what I can tell you is that we are using 3 different pieces, to these glue things and split inteins. We found that they are highly specific. They can't cross-react with other, to generate other toxic proteins. They are highly efficient and highly specific.
Thank you. Thank you.
Yes. Yeah. But first, do you want to use it? Yeah.
So these pieces of the DNA or RNA that we are working on, they have some pairs that it's at the end of one piece, and the half of this pair is at the beginning of another piece. So when these 2 pieces are in the same cell, this pair will find each other. They are like a husband and a wife. So they will definitely find each other in a crowd. So, and they will attach to each other, those pairs that we are using are added as an end and beginning for each those 4 pieces of the genes. So they definitely find each other.
Do you want to talk about degrans? Degrans, the degraded,
Oh, yeah.
the diagrams or?
Yeah, so another also, thank you, Mike.
Yeah.
So another technology that we are still testing is when a cell gets only 2 pieces, not the third, not the fourth, those 2 will be quickly degraded because we don't want them to be, without the 4, all other elements. So what we did is we are trying to include some degradation sequences called degrons. These are present in all natural proteins, and with that we need to help these proteins, if they don't get together, to quickly disappear and get degraded. That's to enhance the safety of this technology. Of course, this is in theory. We need to, we are testing it on cells. It's working great, but is it going to have the same efficacy in mouse? Because it's a complex organ, it has so many organisms, it has so many organs. That is what we will be testing during this grant, thanks to the money from the foundation and for the donor also.
Yep, Macy.
Thank you guys so much, and for the pictures. I probably retained like 2% of that, but that was really exciting to hear. So clearly you guys are specializing in recessive inheritance of RYR-1. Does it matter what variants of recession you have, or could this affect any variant? Like my son has a nonsense and a deletion. Could the AAV still work for something like that, or does it have to be a specific kind? And if so, which variants are you guys using in your grants?
So the mouse model that we will be testing this disease, this approach will be the TM. So it has a missense mutation plus a deletion. So
Okay.
it has 2. The beauty about the gene therapy is that it's a universal approach. So it can be applied to any patient that does not express
Great.
ryanodine receptors, so
Yeah.
recessive forms. This is in theory. We need to apply it, test it in different, models like cells and other, mice also to see if it's the case or not in vivo.
Cool.
So, yeah.
Great.
Just to add on to, in theory, as Haichum just said, the approaches that Afruz, Celine, and Haichum are working on, in theory, and what we expect, that it would not matter what recessive variant you had or your child had, that you are basically, in various different strategies, ultimately trying to give a normal copy of the gene to the muscle cells in the patient. And therefore, it doesn't matter why your child has a lower expression of protein or abnormal, you're giving the patient normal protein. And so therefore, it should be quote, like agnostic.
Yeah, cool. Thank you so much.
Another question over there? Marcy.
Hi, I want to thank you all first for the amazing research that you're doing and taking such a strong interest in RYR-1. I guess my question with the gene editing model, you talk about testing on the cellular level. So my first part of that is what do you see in the cell if you know it is working? Like, for example, in central core, are the core's gone? Or how are you able to identify that it is working, I guess, is part one. And then part two is, in an ideal world, when it is working, let's say, that's my kid right there. Let's say it's working and it's, all the cells have been integrated with this gene editing. Is it instant that things just start, it's like the train stops and restarts again? Or exactly how do you anticipate this process working within the cells and then within the body?
So, just technical observation. It's not gene editing. Gene editing when you modify the
I'm
gene.
sorry.
Yeah, but these 3 approaches, they will be considered as gene replacement. So, for example, for my case, we gave these elements, the 4 elements, to Bob Dirksenson, and he tested this in RYR-1, believe cells, and he saw that the calcium activity was restored as it was with a normal cell. So we believe that it's functional. Now the challenge is to get enough in the muscle, and it's a delivery problem. That's why we are using novel AAVs that I mentioned earlier. These are called myotropic AAVs. They prefer to go to the muscle and not to the spleen, not to the liver, so they have the specificity of delivering the message specifically into muscles. And the second question, sorry, was...
I guess if it is working
Uh-huh.
and it's been delivered, at what point, like, does the body then, like, reboot itself, or like, on what level does that work? Is it, a few muscles here and there, or does everything try to start itself over? How would one day, like, all of a sudden the muscles that help him stand, like, start working?
Yeah, so I would say the positive point about, if you compare RYR-1 related myopathies to muscular dystrophies patients, they lose muscle mass with time, which is not the case. I'm not a clinician, but this is what I heard from my colleagues. So muscle is there, it's just not working properly. So we think that by giving what is missing, the protein that is missing, there will be positive outcomes. We don't know at which level, when, how long does it take. We have, the best example is myotubular myopathy that you showed just the video. So I think it's encouraging, but this is a completely separate disease. I believe that RYR-1 it will be also as efficient and I hope that this will be, the case, but we need to test it. Luckily, we have a mouse model that was created by Jim Dowling and Bob Durkinson. And this mouse model have a short lifespan. They die like after a few weeks, they are born. So we can test this approach or these different approaches in this mouse model and see if we can extend the lifespan of these mice. So that will be a first win. If we can extend them by weeks, months, years, that means that the therapies are working. Then we can do other tests, like we can measure the muscle force, ex vivo or in vivo. We can check the muscle histology by, we collect muscles, we section them, we stain them, and then we see if we restore these core or, central nucleation, et cetera, et cetera. So there are plenty of all these assays and tests that we put in the grants. That I think that's why we got the grant, because they thought, okay, that probably gonna be doable, and that's what we are planning to do.
Thank you so much.
He's correct. Carla.
Yes. I guess kind of to piggyback off of Marcy's question, I know we're not at human testing for any of these things yet, but have you guys had the discussion about how many treatments are going to be required for these things to take effect in a patient? Is it going to be like a one and done thing that you're aiming for, or is it going to be every 6 months and once every year? What is the goal for your treatment?
So I can leave also, my colleagues to talk, but AAVs, a single, if we deliver one gene, just small gene like myotubular myopathy, you saw this video. Mm-hmm. This patient with myotubular myopathy was going to die probably before reaching age of 2 years, and he now has probably age of 8. And what we know also that AAVs, once they are correctly delivered, they express transgenes for more than 11 years. Some data are showing 20 years. These are in animal models. We have, we don't have the same studies in human because, of the challenges with this kind of gene therapies. But we believe one shot will be possible to have expression, a long-term expression for years.
I will repeat the same thing that Hisham said. And gene therapy is one time and a done. So that's one-time treatment, basically. That's the theory behind doing this AAV gene therapies. So one-time delivering of the virus carrying a healthy gene, and it will produce the protein for lifelong. Mm-hmm. So we had patient at Nationwide Children's Hospital, an institute that I'm working on, that has been treated about 10 years ago. Patient was only 6 months old and received the AAV gene therapy for SMA, and now she is about 10 years. So that patient, unfortunately, is supposed to die at the age below 2 years old. And now she is about 10 years old, and she's running, she's a happy girl, and doesn't show any disease phenotype. So it was only one time, and she doesn't need any repeated treatments. So again, that's a concept behind the gene therapy. And for answering a previous question, so how we understand the gene therapy work, so Hisham just talked about there are various experiments that we can do from looking the muscles, functional assays, and other things like lifespan. But gene therapy can prevent a progress of the disease. That's one outcome. or even reverse the phenotype. So two things can come with the gene therapy. And if we are lucky, we have both of them. Like we will prevent the progress and reverse the phenotype and the damage that has been happen in the muscles.
So the hope is that once you deliver the corrected protein, the corrected gene, the body will take over and continue to reproduce it.
Yes, so this gene will remain in our cell nuclei as an episomal plasmid, as a tiny piece of the DNA, remains in the nuclei for many, 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 many years, particularly for muscles, because muscles are already mature cells and they will not divide. So this nuclei will not be divided. The gene will stay in that nuclei, in that cells forever and produce that protein.
Okay, thank you.
So we're a little bit over time. We're gonna do a panel, and so there will be opportunities for more questions, and it'll be very much. Quick question.
Quick question.
Yeah.
Sorry. Yeah, I mean, I know with DMD and all, we have a lost protein there, and gene therapy is perfectly fine for that. So whereas with RYR myopathies, we have a defective protein there. On the other side, we are inducing a new protein. So how they're going to compete? On one side, your body cells, they produce their defective protein. On the second side, we are delivering entirely a wild type perfect protein. So how are they going to compete each other to, for the functional stuff?
I think some patients, they have sometimes, copies of one that is defective and in one functional, and they don't show any disease. So what we think, because this is tetramer, protein, that we will increase the chances of making a tetramer with, correct copies. But this is also, theory. We need to test it and see it if it's the case in mice after AAV injection or, HSV.
All right. All right, how about a round of applause for Haicham, Hafruz, Celine? I hope you appreciate how lucky we are as a community to have people like these researchers working on RYR-1. I have seen all of them present at other meetings on topics unrelated to RYR-1, and I reached out to them. They were all very responsive, and that has continued. They applied for grants. They were reviewed in a competitive process and were awarded grants. And so that we have these kind of all-stars from these other fields now thinking about RYR-1 gives me a lot of hope, and it should give all of you a lot of hope. Really lucky to have these people working on RYR-1. And I would just say, if you want to see more of this kind of work, please support the foundation. The QR code's on the table, but again, none of this would be possible without funding from the foundation. And of course, all of that comes from our generous donors, many of whom are here right now. So if you're interested in funding this kind of work, please support us, and we can do more of it. So once again, thank you so much to you guys. Incredible. Awesome.